Bismillahir Rahmanir Rahim. I'm Dr. Asma Hassan. This is the first lecture in the endocrine system. We will start by the pars distalis part of the pituitary gland, which is the master of all endocrine glands. By the end of this lecture, we should be able to describe the histological structure of different endocrine glands. And we must know the light and the electron microscopic structure of the cells of the pars distalis part of the pituitary gland. And we must correlate the structure of hormone secreting cells, whatever the hormone protein in nature or steroid in nature, to their function. And we must mention how the hypothalamus affects the pars distalis part of the pituitary gland. Endocrine glands. Endocrine means that these glands have no ducts. These glands have no ducts, and their secretion passes directly to the blood through fenestrated blood capillaries. So the cells of these endocrine glands must surrounded by fenestrated blood capillaries to take the secretion directly to the blood circulation. And these cells have no ducts. We have many types of endocrine glands. Pure endocrine glands or mixed glands and solitary cells. Pure glands means that these glands are purely endocrine. No ducts in these glands, as the pituitary gland and thyroid gland. Pituitary gland secretes hormones directly in the blood and has no ducts. Also, thyroid gland secretes thyroid hormone directly into the blood, and this gland has no ducts. Mixed gland mean that these glands have endocrine part and exocrine part. As the pancreas. Pancreas contain part called islets of Langerhans, which secrete hormones directly into the blood, and contain also the pancreatic acini, which secrete enzymes through ducts to reach the duodenum. Secrete digestive enzymes through ducts to reach the duodenum to help in digestion. So pancreas contain endocrine and exocrine parts. Ovaries also contain exocrine part and endocrine part. Endocrine parts that secrete the hormones directly in the blood, and exocrine parts that secrete ova. Solitary cells. Solitary cells means that these cells scattered between the cells of the GIT and lungs. These cells considered as endocrine cells, as these cells secrete directly into the blood, the fenestrated blood capillaries surrounding these cells, not in the lumen. So it's called enteroendocrine cells. These called the cells, not glands, as these cells scattered between cells in the GIT and the lung. So again, endocrine glands means that these glands have no ducts and their secretion pass directly to the blood. There are pure glands, mixed glands, and solitary cells. Hormones that secreted from endocrine glands are different in their nature. These hormones may be protein in nature, amino acid, or steroid. The hormones which are protein in nature as growth hormone, insulin, prolactin, and parathyroid hormones, and the proteins which are amino acid in nature as thyroxine, adrenaline, and noradrenaline. The cells that secrete these two types of hormones are called protein secreting cells. How these cells adapted to make this function or to secrete these protein hormones. The electron microscopic picture of these cells are specific to protein secreting cells. These cells must contain a well-developed rough endoplasmic reticulum, Golgi complex, mitochondria, and secretory granules. As we mentioned in the first year, the rough endoplasmic reticulum responsible for secretion of protein, and Golgi complex make modification to this protein, and mitochondria give energy to this process, and secretory granules that contain or store these protein hormones inside. So the electron microscopic picture of protein secreting cells: well-developed rough endoplasmic reticulum, well-developed Golgi, mitochondria, and secretory granules. The hormones which are steroid hormones, that its precursor is the cholesterol, as the cortisone, aldosterone, estrogen, progesterone, and testosterone. The cells that secrete these hormones adapted to this function. The electron microscopic picture of these cells: will develop the smooth endoplasmic reticulum, mitochondria, but the mitochondria of these cells is specific, as it contains tubular cresti, mitochondria with tubular cresti, and lipid droplets. As the steroid hormone, the precursor of steroid hormone is the cholesterol. So these cells must contain a well-developed smooth endoplasmic reticulum, which secrete lipids, and mitochondria with tubular cresti. Tubular cresti to increase the surface area for the function of the mitochondria. As the mitochondria in these cells, plus it gives energy to this process, it also contains enzymes that help in steroid hormone synthesis. These enzymes present in this cresti. So the mitochondria must contain tubular cresti and lipid droplets, also to store these steroid hormone precursors inside. So we must to differentiate between protein secreting cells and steroid secreting cells, and we must correlate between the function of cells as a protein secreting cells and their structure, and the steroid secreting cells and their structure. Pituitary gland. Pituitary gland is the first gland which we're going to study in the endocrine glands. Pituitary gland called master endocrine gland. Why it's called master? It's called the master endocrine gland as it regulates the function of other glands. As you see, this is the pituitary gland, and it regulates the function of other glands. So it's called master endocrine gland. Its site: pituitary gland lies in the base of the skull. At the base of the skull. And its structure is formed of cords of cells that secrete hormone, and fenestrated sinusoidal capillaries to take these hormones directly in the blood. These sinusoidal capillaries must be fenestrated to facilitate the entrance of the secretion of these cells to the blood. Again, pituitary gland, called master endocrine gland, as it is regulate the function of other glands, and it lies in the base of the skull, and it's formed of cords of cells, and fenestrated sinusoidal capillaries, as it is endocrine glands. Parts of pituitary gland. Pituitary gland formed of two parts, adenohypophysis and neurohypophysis. Adenohypophysis and neurohypophysis. This is the pituitary gland. All this part is the pituitary gland, formed of this anterior part called the adenohypophysis and this dark brown part or posterior part called neurohypophysis. Adenohypophysis formed of three parts. This part, this most anterior part, called pars distalis, and this part called pars tuberalis, and this middle part called pars intermedia. So adenohypophysis called pars divided into pars distalis, pars tuberalis, and pars intermedia. And this dark brown part called the neurohypophysis. It's formed of this narrow part called the infundibular stalk and this wide part called pars nervosa. Again, pituitary gland, which is called hypophyseal gland, formed of two parts, adenohypophysis and neurohypophysis. Adenohypophysis is the anterior part formed of pars distalis, pars intermedia, as it's located in the middle part between pars distalis and pars nervosa, and this part which turn around the infundibular stalk, which is called the pars tuberalis, and this posterior part called the neurohypophysis. What is the parts of neurohypophysis? This narrow part called the infundibular stalk and this wide part called pars nervosa.
Pituitary gland development. Pituitary gland formed of anterior dark cellular part called adenohypophysis and posterior pale part called neurohypophysis. This part is the anterior part, the development of anterior part or adenohypophysis, and this is the development of posterior part or neurohypophysis. Adenohypophysis developed from evagination of the roof of the mouth cavity, evaginated and still evaginated to form in the center a pouch or cavity called the rhatex pouch. Then this part separated completely from the roof of the mouth to form the adenohypophysis part of the pituitary gland. And the neurohypophysis developed from down growth from the floor of diencephalon. Down growth from the floor of diencephalon. This down growth not separated completely from the floor of diencephalon. It is still connected to the brain or connected to the hypothalamus. This part called the neurohypophysis, which is still connected to the hypothalamus. In this part, there is relation between the hypothalamus and pituitary gland through this neurohypophysis. So again, adenohypophysis developed from evagination from the roof of the mouth cavity. This evagination separated completely to form this green part, which is called the adenohypophysis or anterior part of pituitary, and neurohypophysis developed from down growth from the floor of diencephalon, which is still connected to it and called the neurohypophysis. This yellow part called the neurohypophysis or posterior part of pituitary gland. There is another division to parts of pituitary glands, not adenohypophysis or neurohypophysis. We can divide parts of pituitary gland to anterior pituitary and posterior pituitary according to presence to anterior to the rhatex pouch or posterior to the rhatex pouch. Anterior pituitary include pars distalis and pars tuberalis only, as pars intermedia lies posterior to the thoracic pouch. It is related to the posterior pituitary. So posterior pituitary include pars intermedia, pars nervosa, and infundibular stalk. So we can divide parts of pituitary gland as adenohypophysis, which include these three parts, and the neurohypophysis, which contain these two parts, or anterior pituitary contain the parts lie anterior to the thoracic pouch, pars distalis, pars tuberalis, and posterior pituitary, which contain parts which lie posterior to the thoracic pouch. They are pars intermedia, pars nervosa, and infundibular stalk. Pituitary gland is any parenchymatous organ. It's formed of stroma and parenchyma. Stroma, which form the external and internal shape of the gland, and parenchyma is the functioning part of the gland, which contains cells that secrete hormones. So it is any parenchymatous organ formed of stroma and parenchyma. Pars distalis part of pituitary gland. Pars distalis part form the most of pituitary gland. It forms 75% of pituitary gland. These pars distalis formed of cords of cells and fenestrated blood capillaries in between, as these cells secrete hormones which are directed through these fenestrated blood capillaries to the circulation. As any endocrine gland, it's formed of cells that secrete hormones and fenestrated blood capillaries to facilitate the transport of these hormones to blood circulation. Cells in the pars distalis divided into chromophil or chromophobe. Cells of pars distalis may be chromophil or chromophobe. Chromophil form 50% and chromophobe form also 50%. Chromophils means that these cells like stain or have affinity to stains, whatever this stain, acidic or basic stains. And the chromophobe have no affinity to stains, so it's called phobe. It means these cells dislike stain or have no affinity to stains. And the chromophil have affinity to stain. If these cells take acidic stain, it's called acidophil. If they take the basic stain, they called basophils. So the cells in the pars distalis may be acidophil or basophil. They are chromophil or have no affinity to stains called chromophobe. Cells in pars distalis divided into chromophils and chromophobes. Chromophils 50% and chromophobes 50%. Chromophils means that these cells have affinity to stain, and chromophobe that these cells have no affinity to stains. If the granules that store hormone inside these cells stained by acidic stain, it's called the acidophils. And if these granules are stained by basic stain, it's called basophils. So the chromophil divided according to the granules inside. If these granules have affinity to acidic stain, these cells called acidophils. And if these granules have affinity to basic stains, it's called basophils. Chromophobes means that these cells have no affinity to stain, so these cells appear pale by light microscope, appear pale small cells. This is light microscopic picture to the pars distalis of the pituitary gland, showing that these cells, that the cells of pars distalis are chromophobes, small cells, small pale stained cells, as these cells have no granules or few granules inside, not stained, or acidophil, which have affinity to acidic stain, acidic in color, and basophil, which have affinity to basic stains. These cells are arranged in cords and separated by fenestrated blood capillaries. Fenestrated blood capillaries that carry hormones secreted by these cells directly to the blood circulation. Chromophobes, which form about 50% of the cells in the pars distalis, have no affinity to stain, so they appear pale in color, appear as pale small cells. This is light microscopic picture showing the chromophobes. By light microscope, they appear as groups of small rounded cells, pale cytoplasm, central rounded nucleus. Small cells with pale cytoplasm and central rounded nucleus. These cells lies near the center of the pars distalis. By electron microscope, these cells have few secretory granules or no granules, as these cells have no affinity to stain, as they have few secretory granules or no granules inside. What is the function of these cells? These cells with no secretory granules. So it may be undifferentiated cells, undifferentiated stem cells that give rise to chromophyle, or maybe support to other cells. And the few granules which present inside represent the degranulated phase of secretion. Chromophylls. Chromophylls, which form about 50% of the cells in pars distalis and have affinity to acidic or basic stains. These cells lie near the blood capillaries. As these cells secrete hormones, so they lie near the blood capillaries to secrete their hormones directly in the blood capillaries. By light microscope, this is light microscopic picture to the chromophylls. By light microscope, these cells, whatever basophil or acidophil, are oval or rounded cells, and their nucleus central vesicular nucleus. Vesicular nucleus means that this nucleus, active nucleus. These nucleus contain extended chromatin, so appear vesicular with prominent nucleolus. This nucleus, active nucleus. According to the affinity to acidic or basic stains, these cells either basophils or acidophils. By electron microscope, these cells take the picture of protein secreting cells. As these cells secrete hormone protein in nature, so they take the electron microscopic picture of protein secreting cells. What is this picture? As these cells contain well-developed rough endoplasmic reticulum, well-developed Golgi apparatus, mitochondria, and secretory granules. These chromophiles divided into acidophile or basophile. Acidophils form about 35% of all cells in the pars distalis, and basophil form about 15%. But the size of the acidophils smaller than basophils. They more in number but smaller in size, and the basophil less in number but larger in size than acidophils. Basophil form about 50% of cells in pars distalis, but they are larger than acidophils. But acidophils form about 35%, but they are smaller than basophils. The granules inside the chromophil have affinity to acidic stains, as eosine and orangey. But the granules inside the basophils, these granules glycoprotein in nature. Glyco means carbohydrate, means that these cells are strongly PAS positive. But these granules inside acidophil, protein only, not glycoprotein, contain no carbohydrate, so they are PAS negative. This is comparison between the acidophils and basophils. Acidophils divided into somatotrophs and mammotrophs. And basophils divided into corticotrophs, thyrotrophs, and gonadotrophs. Somatotrophs. These cells are the most abundant cells in the pars distalis or in acidophils. By light microscope, these cells appear rounded with acidophilic cytoplasm and the granules stained by acidic stains or with orangey. This is light microscopic picture to the acidophils, and this is electron microscopic picture showing that the nucleus, vesicular nucleus or active nucleus, and the granules, secretory granules, which store the hormone inside. Function of these cells, secretion of growth hormone, or called somatotrophs. So from its name, somatotroph secretes somatotrophin or growth hormone. This growth hormone stimulates the protein synthesis, growth, elongation of long bones at epiphyseal plates. Clinical correlation, acidophil adenoma, case called acidophil adenoma. Adenoma means increase the secretion of the somatotrophs. But if this case occurs before puberty, means that this before the closure of bone. So growth hormone in this case cause elongation of bone, as the bone still not closed. Causes elongation of the bone, this case called gigantism. But if this adenoma occurs after puberty, means that this bone closed. So the growth hormone causes broad bone. This case called acromegaly. So the acidophil adenoma different if it before puberty or after puberty. Before puberty called gigantism, and after puberty called acromegaly. This is a case of gigantism. Decrease the secretion, this case called pituitary dwarfism. Pituitary gland lies under the control of the hypothalamus. Hypothalamus controls the secretion of pituitary gland. Maybe stimulate the secretion or inhibit the secretion of the pituitary gland. So, hypothalamus stimulates the secretion of growth hormone or inhibits the secretion of growth hormone, as the pituitary gland lies under the control of the hypothalamus.
Mammotrophs are the second type of acidophils. Light and electron microscopic picture of mammotrophs is any protein synthesizing cells, but they appear small and spindle-shaped cells in male and non-pregnant female. The function of these cells is the secretion of prolactin hormone. Prolactin hormone released especially or secreted especially in pregnancy and lactation. Why? Because during these periods there is high estrogen level, and the prolactin hormone causes development of mammary glands in pregnancy and milk secretion in lactation. So, in these periods, mammotrophs appears with numerous rough endoplasmic reticulum and well-developed Golgi, and the granules these mammotrophs inside these mammotrophs increase in number and enlarge to triple its size. So during these periods, mammotrophs called pregnancy cells. And this is the characters of these pregnancy cells or mammotrophs during the pregnancy and lactation.
Now we will start in the basophil. As we mentioned, basophils divided into thyrotrophs, corticotrophs, and gonadotrophs. Thyrotrophs are the first type of basophils. By light microscope, they appear as large polyhedral cells with basophilic cytoplasm and vesicular nucleus. And the granules inside is these granules glycoprotein in nature, so they are strongly PAS positive. And the nucleus, central vesicular nucleus. By electron microscope, these cells contain parallel cisternae of rough endoplasmic reticulum, Golgi apparatus, mitochondria, and secretory granules. This is a picture of what? This is a picture or electron microscopic picture of protein secreting cells. These cells adapted in its structure to secrete protein. So they contain parallel cisternae of rough endoplasmic reticulum, Golgi apparatus, mitochondria, and secretory granules. This is the secretory granules inside. The function of these cells is the secretion of thyrotrophic hormone, or which is called the TSH, thyroid stimulating hormone. That stimulates the thyroid gland to secrete thyroid hormone. We mentioned before that pituitary gland lie under the control of hypothalamus. So, hypothalamus secrete thyrotropin releasing hormone, TRH, thyrotropin releasing hormone. This stimulate pituitary gland to release TSH, thyroid stimulating hormone, which stimulates the thyroid gland to secrete T3 and T4, which are called the thyroid hormones. These hormones cause negative feedback on both pituitary gland and hypothalamus to decrease TRH and TSH. This diagram also showing that hypothalamus stimulate pituitary by TRH. Pituitary release TSH, which stimulates thyroid gland to release thyroid hormone, which causes negative feedback to increase hypothalamus hormone and anterior pituitary hormones, or TRH and TSH. Corticotrophs, the second type of basophils, take the same light and electron microscopic picture of thyrotrophs, or protein secreting cells. By light microscope, these cells appear polyhedral in shape, basophilic cytoplasm, and vesicular nucleus. And by electron microscope, these cells appear as protein-secreting cells. Protein-secreting cells, well-developed, rough endoplasmic reticulum, Golgi apparatus, mitochondria, and secretory granules. Secretory granules. The function of these cells that they synthesize the pro-opiomelanocortin. This is a precursor that give adrenocorticotropic hormone, ACTH, adrenocorticotropic hormone, and lipotropic hormone in lipid metabolism, and melanocyte stimulating hormone, which stimulate melanin synthesis. But the main hormone secreted from the corticotrophs is the adrenocorticotropic hormone, which stimulates the adrenal gland to secrete cortisol. These cells lie under the control of pituitary gland. This diagram showing that adrenal gland or suprarenal gland that secrete cortisol stimulated by the pituitary gland, and pituitary gland is stimulated by the hypothalamus. Hypothalamus release CRH, corticotropin releasing hormone, that stimulate corticotrophs in the pituitary to release adrenocorticotropic hormone. This hormone stimulates the adrenal gland to secrete cortisol. And this cortisol causes negative feedback. What is the meaning of negative feedback? Negative feedback means that this cortisol inhibits both pituitary and hypothalamus. Negative feedback means the hormone inhibits the secretion of hypothalamus and pituitary gland. So, corticotrophs stimulated by the hypothalamus and inhibited by negative feedback from the cortisol secreted by adrenal cortex. Gonadotrophs are the third type of basophils. These cells take the light and electron microscopic picture of protein secreting cells as corticotrophs and thyrotrophs. This light microscopic picture to gonadotrophs, and this is electron microscopic picture vesicular nucleus, secretory granules, and basophilic cytoplasm with vesicular nucleus. Function of these gonadotrophs is the secretion of follicular stimulating hormone and luteinizing hormone. Follicular stimulating hormone, which stimulates the growth of ovarian follicles in female, and secretion of androgen binding protein by Sertoli cells in male. And the luteinizing hormone stimulate ovulation and corpus luteum formation in female and testosterone production in males. Gonadotrophs also regulated by the hypothalamus. Hypothalamus release corticotropin releasing gonadotropin releasing hormone, stimulates the pituitary gland to release luteinizing hormone and follicular stimulating hormone, which affect the gonads in male and female. The secretion of gonads or hormone secreted from gonads causes negative feedback on both pituitary and hypothalamus. So these gonads or secretion of gonads inhibits the secretion of the hypothalamus and pituitary. Castration cells. Castration cells is the same gonadotrophs, but after removal of gonads. After removal of gonads, gonadotrophs in pituitary increase in size and become full of secretion as nothing to stimulate, no gonads. So these cells accumulate secretion inside and increase in size and become full of secretion, and this secretion pushing the nucleus to the periphery. This is the shape of cells called the castration cells. It is the same gonadotrophs but after removal of gonads. This is electron microscopic picture to the pars distalis that showing the cords of cells surrounding fenestrated blood capillaries. These blood capillaries contain fenestrate to facilitate entrance of hormones secreted by these cells to the blood circulation. And the cells of pars distalis are chromophobe and chromophile. Chromophile may be somatotrophs, gonadotrophs, thyrotrophs, and so on. But chromophobe contain no or few secretory granules inside. All chromophile contain secretory granules inside. So these cells most probably chromophile and these cells most probably chromophobe. Comparison between acidophils and basophils. Acidophils by the percent are 35%, and basophil 15%. But acidophils are more in number and smaller in size. They are smaller in size than basophil. Basophil larger in size. Acidophils, from the name acidophil by light microscope, it is acidic. It is acidic as it take acidic stains as eosine. But basophil basic stains as hematoxylin. Take basic stains as hematoxylin. By light microscope, they have the same picture. They are oval or rounded cells with eccentric vesicular nucleus. The granules inside are different in nature. The granules inside acidophil are protein in nature, so they are BAS negative stain. BAS negative. But the granules inside basophil are glycoprotein in nature, so they are BAS positive. So when we stain pars distalis by one stain, we can differentiate between acidophil and basophil. This BAS stain. Basophil strongly BAS positive, but acidophil BAS negative. By electron microscope, the two cells are protein secreting cells. So the cells adapted in its structure to this function. They contain rough endoplasmic reticulum, Golgi apparatus, and mitochondria, and secretory granules. But the granules inside basophils are small granules of variable electron density. Acidophils are two types, somatotrophs and mammotrophs, but basophil are three types, thyrotrophs, corticotrophs, and gonadotrophs. Now we have finished the lecture, and this is multiple choices on the lecture. Somatotrophs are chromophobes, acidophils, basophils, or pituithides. The right answer, letter B. Letter B, somatotrophs from the acidophils. Mammotrophs are chromophobes, acidophils, basophils, pituithides. It's also letter B, as acidophils have two types, somatotrophs and mammotrophs. Pars distalis contains somatotrophs and chromophobes, corticotrophs, herring bodies, no, pituithides and herring bodies, no, spongiocytes and supraoptic nuclei. The only cells that present in pars distalis are somatotrophs and chromophobes, so the right answer, letter A. Pars distalis include all the following except pituithides, chromophils, somatotrophs, corticotrophs. We studied all these cells, but pituitide in the posterior pituitary. So, the pituitide, or letter A, is the right answer. During pregnancy, which of the following cells increase in size and number? Normally during pregnancy we need lactation, prolactin hormone. So the cells which increase in size and called pregnancy cells are the mammotrophs. The right answer, letter C. A 30-year-old patient complaining of changes in facial features that became coarse with protruded mandible and enlarged hands. This is due to oversecretion from corticotrophs, gonadotrophs, mammotrophs, somatotrophs. Protruded mandible and enlarged hands means that it's broad bone, not long bone, broad bone. This means that this patient exposed to oversecretion of growth hormone after puberty, after closure of bone, which is called acromegaly. This is due to oversecretion of growth hormone secreted from somatotrophs of acidophils. So the right answer, letter D. Now we have finished the lecture. Thank you. Assalamu alaikum wa rahmatullahi wa barakatuh.